Good evening, everyone. Welcome to, what week are we in now? Week 8 of Living Systems. And tonight we're going to finish last week's lecture, Lecture 10, Solutions. So let us proceed to where we left off. And by the way, this is the last of our chemistry lectures. And then tonight we'll start on our biology component. But of course, covering the biology in the second half of this subject will make lots and lots of references back to what we learned in the chemistry component. So you'll see that there's, you know, such a huge benefit to actually understanding some chemistry in order to have that deeper understanding of the biology. So we finished here last week, and so we'll be continuing on from this slide where we consider the units of concentration. So concentration can be defined as the amount of solute that is dissolved in a given amount of solution. So of course, as you know, there's different units that you can use for concentration. Let's think of some of them that you might be familiar with. Who can give me a unit of concentration?
Molarity?
Molarity, yes. So big M, molarity. And what is that telling us? What is the units of concentration for molarity? Moles per litre.
Yeah, okay.
Yep. So amount of solute dissolved in a given volume. So that is a form of concentration. What about another unit of concentration that you might be familiar with?
Milligrams per litre? Oh, no, that's...
Yeah, that's right. Milligrams. Yeah, milligrams per litre. Absolutely. That's supposed to say milligrams. So milligrams, that's an amount in a given volume. What about something like, oh, here's a good one. What about a ratio? What about 1 to 10? That's also a concentration. Yep, 1 to 10. Or we can say, slightly different, 1 in 10 would be different. So 1 to 10 means 1 part of something in 10 parts of, you know, water or whatever the solvent is. 1 in 10 is a little bit different, isn't it? Mm-hmm. Because this means that you've got 1 part in, or rather 1 part plus 9 parts. So 1 part of the solute, for example, and 9 parts of the solvent, for example, water. So slightly different. But ratios are also considered a concentration. What if you just said milligrams? Is that considered to be a concentration?
It's a weight, but
it is. That's right. It's an amount. It's a weight. But it's not a concentration. It's just telling you that you've got so many milligrams of a substance, but it's not giving you the volume that amount is actually dissolved in. So this is an amount, but it's not a concentration. All right, a concentration that you're probably familiar with now because of the pracs that you've done is weight per volume percent concentration. So this is simply the number of grams of a solute dissolved in a given volume of solution. Weight per volume percent concentration. Pretty straightforward. Weight per volume percent concentration equals the mass of your solute in grams divided by the total volume of your solution. Keep that in mind, total volume of the solution in mil, and then that's multiplied by 100. And that will give you weight per volume percent. Let's look at an example of that. Here we go. For example, vinegar contains 5 grams of acetic acid in 100 ml of solution. So to work out the weight per volume percent concentration, we simply take the solute, 5 grams of acetic acid, divide that by the total volume of the solution, 100 ml, multiply that by 100, and here's the answer, 5% weight per volume. So let's think now about how you would make this up. Because it's all right to actually know how to work this out, but how would you actually make this up, this solution? Okay, here we have some sort of measuring flask. I'm going to use a measuring cylinder. Here we go. We're going to weigh out 5 grams and place it in here. So here's our 5 grams. There we go, 5 grams of your solute. Then we would top that up to the 100 ml mark with water. Top that up to the mark, and there we have our 5% solution. So remember, keep that in mind, this is our total volume, including the 5 grams. Okay. If we actually weighed out that 5 grams and then added 100 ml of water to it, obviously that is not a 5% solution. That would not be correct. All right, our next unit of concentration, very similar, volume per volume percent concentration, where we have a volume of solute in mil, and that is dissolved in a total volume of solution and multiplied by 100. Let's look at our example. If a bottle of alcohol contains 70 ml of the alcohol 2-propanol, in a total of 100 ml of solution, then the volume per volume percent concentration, simply 70 ml of 2-propanol divided by 100 ml of total solution and multiply that by 100%. And that's pretty easy. You can work this out without even doing the math, 70% volume per volume. Now, I just want you to keep in mind for one of the tutorial questions, which is a bit of a trick question. Let's say you had 70 ml of a given, sorry, that's supposed to be 70 ml, 70 ml of a given solute and 100 ml of the solvent, and you're asked to add them together. So it's not 70 in 100, but 70 and 100. That means this number, this total volume changes, doesn't it? Yep. Okay, so that would actually be 70 divided by
170.
170. All right, so look out for this, because there is a trick tutorial question designed just for that. All right, so make sure that you're very familiar with these two, because they always come up in the final exam, calculating volume per volume and weight per volume percentage. All right, our next unit of concentration, molarity, which someone's already mentioned. And you already know that molarity is the number of moles per litre of solution, and we abbreviate that to big M. So there's nothing new that's actually being introduced here, because we've already talked about molarity, and this is simply giving you some revision. So let's look at the example here. Calculate the molarity of a solution made from 20 grams of sodium hydroxide in 250 ml of solution. All right, so because you're being asked to calculate molarity and you're given volume and you're given grams, what formula that you're already familiar with, what formula do you think you would use? Just thinking about that term molarity. Who remembers one formula that we already used in week 6, I believe? Week 5 or 6 you were introduced to it.
N equals CV.
Correct. N equals C times V. Okay, let's revise. What does the N stand for?
Mole.
Number of moles, correct? So that's amount or number of moles. What does the V stand for?
Volume.
Volume in?
Liters.
Litres, yes, it must be in litres to make this formula work, correct? And what about this C?
Concentration.
Concentration, correct, in using the units of? Molarity, yes, so moles per litre. Good. All right, so let's see if we can use that formula to solve this problem here. So we've got our volume, 250 ml, and what do we have to do with that?
Convert it to litres.
Convert it to litres, correct, yes. So we can do that now. Let's convert that. 0.25 litres. All right, we're being asked to calculate molarity. So this is our unknown right here. Okay, and what about here? We need to know the number of moles, but we don't have that. We have grams.
Mm-hmm.
Yep. Now, so we can't use this yet, this formula yet, because we've actually got 2 unknowns. So what do we do when we know grams, but we need moles? What formula can we use?
Mass equals...
Correct. Mass equals number of moles multiplied by...
MR, or molecular weight.
Yes, MR. Yeah, you can say MR. Yep. Molar mass. Remember, this is molar mass. Yep, so this is our molar mass formula. And because molar mass has the units, who remembers the units? Grams per mole. Grams per mole. So this molar mass is used as a conversion factor to relate mole to mass. Yep. All right, so we know mass, 20 grams is our mass, 20 grams. We need to know number of moles. This is our unknown. And where do we get this molar mass from?
From a periodic table.
Correct. So always remember, molar mass you can calculate directly from the periodic table. So let's do that. Get your periodic table out. I'll give you a minute. Get your periodic table because I want you to work out the molar mass of sodium hydroxide. All right, so I'm going to give you a few minutes. So remember, find the atomic mass or atomic weight of sodium, of oxygen, of hydrogen, and then add them all together using the periodic table. But don't give me an answer yet. Making a bit of noise there, someone.
Yeah, just opening up. Got to get a calculator out.
Ah, okay. And just put done in the chat box when you're finished. Okay, for those of you that are already done, I want you to solve this. Okay, very good. All right, let's go back to our molar mass. What did everyone get for molar mass? Okay, we've got 40 and 40. That is correct. 39. That's close. All right, 40 grams per mole. All right, that sounds very good to me. How do you rearrange this equation so that we can solve for number of moles?
It's mass over molecular weight.
Not molecular weight.
Or MR, or it's in
molar mass.
Yeah.
Molar mass. Yeah, number of moles equals mass over molar mass, which equals 20 grams divided by 40 grams per mole. And what do we do before we solve this equation? What should we do? What should we do? What is best practice before we
cancel out the common units?
Correct. We cancel out gram with gram. Yes. We look at the remaining unit, which is mole, and do a quick check. Is that what we're trying to solve for? Yes.
Silvana?
Yes.
We actually can't see the bottom of the screen, that's all.
Oh, right. Okay. Oh, that's interesting.
So I can't see what you're...
All right. I'm going to rewrite it here. Thank you. Thank you, Andrew. Okay. Number of moles equals mass divided by molar mass equals 20 grams divided by 40 grams per mole. And we cancel out the like units. So we've got grams, grams, and what answer did you get?
0.5 moles.
0.5 moles. Correct. All right, so now we know how many moles we have. We can go back up to N equals C times V. And we have 0.5 mole. Oh, that's terrible. Wow. You'd think after years of doing this, I would be really good, but I'm not. All right, so we've got our number of moles, we've got our volume in litres. Now we can rearrange to solve for molarity, which is what the question asked us to do. So I want everyone to rearrange this formula and solve. I'll give you a minute to do that yourself. Okay, how's everyone going? All done?
Yep. Oh.
Yep.
Yep.
Okay, so we rearranged to solve for concentration, molarity equals number of moles divided by volume, 0.5 mole divided by 0.25 litres. We can't cancel out any units there, which is good because we're trying to solve for molarity. So what answer did you get here?
2.0.
2.0 moles per litre, or just big M, molar. 2 molar. Or if you write 2 moles per litre, I would accept that just the same. All right, terrific. So this is a really good practice session for you to do the 2-step calculation using both of those formulas. So as you can imagine, also something that comes up all the time. All right, let me clear all that. And now we can move on. So nothing new here at all. There's the answer, 2 molar. All right, let's look at another example. What volume in millilitres of a 0.3 molar solution of glucose contains 0.025 mole of glucose? What formula shall we use for this problem?
Same one.
Same one.
N equals CV.
Correct. N equals CV. We'll do the job. All right. I'll give you a minute to work this yourself. Okay, how did we go? Yep. Did everyone rearrange to volume equals number of moles divided by concentration?
Yep.
Yes. So which would have given you 0.025 mole divided by 0.3 molar. Now remember to include the units for molarity, moles per litre, which will allow you to cancel mole with mole, and this leaves us with the desired unit of volume, litres. All right, so what answer did you get for this?
83 mils.
83 mils, so you would have, the answer would have been 0.83 litres, which you then have to convert to, 0.083 litres, which you would then have to convert to mil. Is that right?
Yeah.
Everyone happy with that?
Yep.
Okay, terrific. Good. All right. Now, is everyone aware that the mid-semester exam is coming up?
Yep. Sunday.
Yes, Sunday, correct. So remember, it opens this Sunday morning, 6 o'clock, and it'll close the following day, Monday evening at 11 p.m. Okay, so you can start at any time, but once you start, then you'll have to complete within the 2 hours. So don't start at 10 p.m. Monday evening, because it will just close at 11 o'clock. I've seen that happen, so not good.
And which lectures is it?
Are covered only weeks 1 to 5, which includes lectures 1 to 6.
Thank you.
Oh. All right. Let's move on now to dilution. Also not a new concept for you. Dilution we talked about in the very first week of semester, if you might remember. So this is where we actually talk about dilution in this lecture on solutions, but you're introduced to the formula C1V1 early in semester to give you time to practice. Because some students really don't like the maths at all. So C1V1, we use this to work out our dilutions. All right, so I don't think there's any need to go over this, apart from just showing you that the M1V1 equals M2V2, shown here, is exactly the same as our C1V1 equals C2V2. All right, so let's look at an example. So as a bit of practice, I'd like you to work out this problem. What is the concentration of the solution formed by diluting 5 ml of a 3.2 molar glucose solution to 40 ml? Oh, we've got someone very noisy in the background. All right, so I'll let you work on this for a couple of minutes and see how we go. Just let me know when you're done. Okay. Terrific. Okay, so which one is our unknown?
V2. Sorry, C2.
C2.
C2, yes. So you're being asked for concentration, yeah. And this is often the trickiest part for students to actually decipher the question itself. But that's correct. So that's our unknown, C2. what is our C1?
3.2 molar.
3.2 molar is correct. So we've already got this 3.2 molar solution. What volume?
5.0.
5 mil.
5 mil. Yep, correct. And what final volume do we want?
40 mil.
40 mil, correct. All right, good. Now, how did you rearrange this to solve for C2?
To get C2 on its own, you divide by
Yep. Sorry, I didn't catch that. C2 equals?
We'll divide by V2 so that they cancel out.
Yes,
And so it's C1 V1 over V2.
correct. C1 V1 over V2. Yes. Yep, so we've got 3.2 molar. 3.2, I'll put moles per litre, multiplied by 5 mil. Divided by 40 mil. Okay. All right, so can we cancel units? Yes, we can. Cancel. Cancel. We're left with moles per litre. Is that correct? Yes, because we're trying to solve for concentration, and we already have concentration using molarity. So what was your final answer there? What did everyone get?
0.4 molar.
Did everyone get the same?
Yep.
0.4 molar. Yes?
Yep.
Okay. Good. All right. That's terrific. So just some more revision for you there. All right, nearly done with this lecture. We're just going to finish very briefly with a look at colligative properties, which are properties of a solution that depend on the concentration of the solute, but not its identity. So the number of dissolved solute particles affects the properties of the solution, but the identity does not. Colligative properties includes elevation of boiling point, lowering of melting point, and the pressure due to osmosis. We're not going to talk anymore here about osmosis, but we will visit this in a later lecture. So let's start with boiling point elevation. To consider this, we need to think about solutes in solution and whether they're volatile or non-volatile. You're probably all familiar with the fact that a volatile solute is one that readily escapes from the liquid phase into the vapor phase, whereas a non-volatile solute does not readily escape into the vapor phase. And because it doesn't readily make that transition into the vapor phase, that means this solution has a very low or negligible vapor pressure at a given temperature. So the vapor pressure above a solution of a non-volatile solute is lower than the vapor pressure of the pure solvent alone. So let's look at how this contributes to elevation of the boiling point. So how do we normally define boiling point of a solution? Sorry, what determines the boiling point of a solution? That is the temperature at which a solution boils. What determines that? Who remembers?
Goes from a liquid to a gas.
Yes, it's certainly, we're talking about transition from liquid phase to gas phase. But what is it that actually determines the temperature at which that occurs?
Oh, is it something to do with the breaking of the ionic bonds or something like that?
Yes, and remember it's also has to do with the actual vapor pressure itself. Yeah.
Oh, the pressure inside the vessel, or
The pressure are just above that solution. When that pressure just above that solution equals atmospheric pressure, then there's enough energy there to cause that liquid to boil. So that's something that we visited briefly in a previous lecture. All right, so boiling point elevation means that we have a liquid solution contains a non-volatile solute, and this will have a higher boiling point than the solvent alone. The amount that the boiling point increases depends only on the number of dissolved particles. So if we have 1 mole of a non-volatile solute and we place that 1 mole into 1 kilo or 1 litre of water, then the boiling point will increase by this temperature, 0.51 degrees Celsius. Let's try to visualise that. Let's say you had a pot of water. Pot of water, we had 1 litre of water in that pot, and we placed, and normally that water would boil at what temperature?
100.
100 degrees, yep. If atmospheric pressure was 1 atm, then the water would boil at 100 degrees Celsius. But let's say we placed 1 mole of a non-volatile solute. For example, salt, sodium chloride. Let's put 1 mole of sodium chloride. Yep. This would cause an elevation of the boiling point from 100 degrees Celsius to 100.51 degrees Celsius. Okay, so we've elevated that boiling point, elevated that temperature. All right, is that why we put salt in water when we're cooking pasta?
Yes.
I don't know. I was actually asking the question. There you go.
So
Thank
it
you.
cooks faster, I think, isn't that's the...
I don't actually know. I think I always thought it just had to do with taste, but I'm wondering if there's a bit more to it that I don't know about. Yeah.
Liz with that. I think it elevates the water temperature too.
Yes. So you want to cook rice or pasta in water that's as hot as possible, I suppose. Yeah, I don't know. But anyway, let's clear this. Oh, it's no good. Oh, and here we go, last slide. So here we're looking at the same sort of thing, but depression, this time depression of freezing point. Let me clear that. Okay. Freezing point depression. So a liquid solution that contains a non-volatile solute will have a lower freezing point than the solvent alone. So let's say that we freeze water, just pure water. What is the temperature at which it freezes?
0.
0 degrees Celsius. But if we place 1 mole of a non-volatile solute into that water, then the freezing point will actually decrease by 1.86 degrees Celsius. So if you consider the freezing point of pure water, and imagine we add something, a non-volatile solute such as antifreeze that we put in the radiator of the car. Yes, antifreeze, that means the freezing point of water will then be negative 1.86 degrees. Yep. Does that make sense? And we've got a very nice example here. The Antarctic ice fish produces its own antifreeze in the form of glycerol. And here's the chemical formula for glycerol, and this means that its blood, its body fluids, actually remain fluid even in really cold temperatures that would normally freeze liquid. So I think a nice example to illustrate that.
I think I preferred your food analogy better. So.
Oh, do you? You don't like this one?
No, that's right.
I think this one's even easier to understand.
Sugar and water and moisture is a good one.
We're all
Right.
hungry.
Oh, I'm making you hungry. Don't worry, I haven't had dinner either. And my daughter's cooking pasta, so I'm feeling really hungry. All right, let's move on to the first of our biology lectures, functioning cells. Move that over a bit. So students sometimes say, oh, great, biology, it's really easy. But biology is very different in terms of thinking processes and the way that you have to learn, because it is so descriptive. A lot of new terminology, a lot of memory work involved. So sometimes students say, oh, I wish we were still doing chemistry. But anyway, we'll see how we go. We have an amazing image here of plant cells. Really nice. Some of you might have seen some great cells through the microscope during your prac workshop. And some of you may have seen that it takes practice. If you've never really used a microscope before, or you've got very little experience with a microscope, it's not something that you can pick up within a couple of hours. It takes lots and lots of practice to be really, really comfortable with using a microscope, and to be able to use it in such a way as that so that you don't get a sore neck, or you don't get sore eyes, you don't get a headache, and so on. That just all comes with practice and knowing how to work the microscope to suit yourself, to suit your posture, your eyes, and so on. All right, so functioning cells. Before we start with this lecture, let's consider what it means to be living. Living organism. What is it that every living organism on Earth would have in common? Sounds like a really easy thing to define. Living. What do all living organisms on Earth have in common? Any idea?
The ability to metabolize food into energy?
Yes. Well, you would think so. And most living organisms do have that ability, but there are some that actually don't. There are some parasitic bacteria, for example, that are become so specialised at being parasites that they can no longer metabolize fuel molecules to make energy. They depend on the cells that they parasitize to actually get their ATP. So yeah, really, really good point, and that would apply to just about every living organism, but not quite. So that's actually a really good illustration of how difficult it is to define living. Some people say the ability to reproduce, but as you know, there are organisms that are sterile. They can't reproduce. You know, hybrids, for example, they have no way of reproducing. We can say, okay, the ability to move, but we know that there are sessile organisms that don't move. They always just stay in one spot. So it's a little bit more complicated, but I think what's called the cell theory, this is a theory that was devised over the last 250 years or so, actually covers this concept quite nicely. Cell theory. Cell theory Yes, go on.
Is that living is something that consists of one or more cells, or two or more cells?
Correct. That's it. That is one part of cell theory, which states that all living organisms are made up of cells. All living organisms are made up of cells. And you won't really find any exception to this rule. That's number one. Number two, under the cell theory, is that all cells come from pre-existing cells. So cells don't just appear out of nowhere. They have to be made by cells that are already existing. So please don't ask me where the first cell came from. I do not know. Nobody knows how the first cell arose. There's some interesting theories on how the first cell came about, but we certainly won't go there. But you can look that up if you want to. All right, number three. The cell is the basic unit of life. Where we saw that the atom is the basic unit of matter, of all matter, the cell is the basic unit of life. So that raises the question, how do we define a cell? How do we define a cell so that it's distinct from the cell in your car battery? Yep, we're talking about a biological cell. We can say very simply that a cell can be defined as a very small compartment enclosed within a biological membrane, and we'll talk about membranes, where biological reactions take place. That's a nice, simple definition of a cell. All right, so let's clear all this. Now, there are two major classes of cells. Prokaryotic, and you know all this because you've done the pracs. Prokaryotes, and what's the other one?
Eukaryote.
Eukaryotes, correct. Eukaryotes. And what do these terms actually mean? Eukaryotes. Where are they derived from, these terms? So so this term here is derived from prokaryon, which means before, and my writing's getting worse as I'm getting more hungry, before kernel, or before the nucleus. Kernel or nucleus, I'll come back to what that means in just a second. Eukaryotes, you meaning true or proper kernel. True or proper kernel. So what that's referring to is this distinction between a eukaryotic cell, which contains this structure, and this is the nucleus of the cell. Okay, so the kernel is referring to the nucleus. Eukaryotic cells all contain a membrane-enclosed nucleus. A prokaryotic cell, on the other hand, lacks a membrane-enclosed nucleus. And that's because prokaryotic cells are a more ancient type of cell. They're thought to have been the first living cells that evolved on Earth. That is bacteria. And does anyone know another group of prokaryotes? There's bacteria.
Amoebas?
No, not amoebas. Bacteria is one group of prokaryotes, and there's one more group, and they're called archaea. They're very similar to bacteria, but they have some differences, and hence they're in a different group here, archaea. So these are the only two groups of prokaryotic organisms. And what about eukaryotes? What groups of organisms are eukaryotic organisms?
Plants.
Plants.
Animals.
Animals, correct. Plants. Animals, they're all made of eukaryotic cells. Yeah. What about fungi? Fungi are also eukaryotes. And there's one more group. Protists. The protists. So this is a group that's really quite varied in the organisms that are found here. But one group found in the protists you'll be familiar with, algae. Algae belong to this group. Algae. Okay, there you go. So, main point here. Eukaryotic cells always contain a membrane-enclosed nucleus. What do we find in the nucleus?
Chromosomes and DNA.
DNA, yeah. Yep. So the nucleus houses and protects the DNA of the cell. Do prokaryotes have DNA? Well, of course they do. All living cells have DNA, but the DNA, which I'm trying to draw here, is not enclosed in a membrane-bound nucleus. Okay, eukaryotic cells are also a lot more complex in that they often, not always, but often contain other structures. And these structures, many of these structures, are also enclosed in biological membranes. You don't find them here. You don't find membrane-enclosed structures at all in prokaryotic cells. They do contain some structures, but not membrane-enclosed structures. All right, so we're going to start with our eukaryotic cells. Eukaryotic cells contain membrane-bound structures which are called organelles. For example, they always contain a nucleus which houses the DNA of the cell. Another example of an organelle that is a membrane-bound structure is the mitochondria, and here's another example, the endoplasmic reticulum. There are more examples of organelles which we'll come back to. All right, structures that are not membrane-bound. Ribosomes, and you'll also find ribosomes in prokaryotic cells, and also microtubules. So just two examples shown here. Excuse me. The aqueous solution within the actual cell is called the cytosol. And there's another term here, which I'll just write down for you. And it's called the cytoplasm. Sometimes people use these two terms, cytosol and cytoplasm, interchangeably, but the cytoplasm is a little bit different. It is referring to that aqueous solution inside the cell, but also all the structures that are found within that solution, except for the nucleus. All right, here we have a typical animal cell, and it's been cut in half to show the contents, and you can see that it's a very busy place inside this eukaryotic cell. All right, in here in blue and also partly cut away here is the nucleus, and we have this dark staining region inside the nucleus called the nucleolus. In brown, we have the endoplasmic reticulum, also an organelle. Another organelle here in purple, the Golgi apparatus, or Golgi complex. These little yellow organelles, peroxisomes. These organelles, lysosomes. Over here, you can see all these tiny little black dots. These are called ribosomes. and ribosomes are also found in prokaryotic cells. And then we have components of what's called the cytoskeleton. So essentially a skeleton or scaffolding within the cell. And you can see these filaments here. They're referred to as microfilaments. And we have other filaments over here, also part of the cytoskeleton, microtubules. All right, all cells are enclosed in this biological membrane, and this is referred to as the plasma membrane. So every living cell is surrounded by a plasma membrane, regardless of whether it's prokaryotic or eukaryotic. So let's take a look at membranes. So we've talked about membranes in a previous lecture. And as we just said, membranes enclose both the cell, and this is called the plasma membrane, but membranes also enclose certain structures inside the cell, and they're referred to as organelles, which actually means little organ. So it's almost like the cell is its own entity, its own complete living organism with these little organelles. Now, you know that membranes, biological membranes, consist of a lipid bilayer of phospholipids. And if you can't quite remember the phospholipids, you might remember that we draw them simply like this. This is one phospholipid. Here's the polar head, which is hydrophilic. And you know that means that it likes to associate with water. And then we've got these two tails. These are fatty acid tails, and they are hydrophobic. So they are water-hating or water-fearing. Okay, membranes are also, they're primarily composed of phospholipids, but they also have lots of proteins attached to them or embedded within them. Those proteins are broadly classified into two groups: the peripheral membrane proteins, which are attached very loosely by non-covalent interactions, and the integral membrane proteins. And many of these integral proteins are transmembrane. That is, they extend right through that lipid bilayer of the membrane from one side to the other side. And we'll see this on the next slide. Here we go. Here's our portion of a membrane. And why is that not clearing? Oh, here we go. All right, so let's look at this membrane. Let's dissect it. So here we have our phospholipid bilayer. Okay. Here we have an example of a peripheral membrane protein. It's just attached to one side. Oh, by the way, this side of the membrane down here represents the inside of the cell, and this part up here, the outside of the cell. All right, here we have an example of an integral membrane protein, and because this one extends right through the lipid bilayer, it's called a transmembrane protein. These are important because they normally contain a pore, a channel, that allows certain substances to move from the inside of the cell to the outside, or vice versa. Here's another embedded membrane protein, also an example of an integral membrane protein, but not transmembrane. All right, if we look at the outside of the membrane, we can see that some proteins are attached to a carbohydrate chain, and they're referred to as glycoproteins. Then also on the outside of the membrane, you might have a carbohydrate chain here, which is attached directly to the phospholipid bilayer, and these are referred to as glycolipids. All right, so we've seen that membranes usually have different molecules on each side. They're not symmetrical structures. We also saw that carbohydrates may be attached to lipids, which are then called glycolipids, or the carbohydrates might be attached to proteins, which are then referred to as glycoproteins. The carbohydrates normally occur on the non-cytosolic side of the membrane. Okay, so in other words, on the outside of the actual cell, if we're talking about the plasma membrane itself. If we're referring to the membrane surrounding the organelles, then the carbohydrates occur or are found on the inside of the membrane, that is facing towards the lumen, the inside of the actual organelle. But as we just said, the carbohydrates are found on the outer surface of the plasma membrane of the cell, and it forms a layer called the glycocalyx. So many new terms. All right, so properties of membranes can be summed up right here with these two words. Membranes are said to be fluid mosaics. They're referred to as mosaics because of that asymmetrical nature of the membrane. That is, we have proteins and carbohydrates embedded or attached in an irregular arrangement that is different on one side of the membrane compared to the other. So that's the mosaic part. Membranes are said to be fluid because the lipids within the membrane and some of the protein molecules can actually move laterally. So a membrane isn't just a fixed structure. It has quite a fair bit of fluidity about it. Membranes also have another important property. They're said to be selectively permeable. They are permeable to some substances, that is water, oxygen and carbon dioxide can freely cross through that lipid bilayer of the membrane, can just freely move across from one side to the other. Most substances, for example, charged species, that is ions, and any other polar molecules can only cross at selective pores that we find within those transmembrane proteins. Now that sounds odd that we're saying polar molecules because water is a polar molecule and yet it can just freely cross through the lipid bilayer. How water molecules do that, no one's really sure. But they certainly do cross directly through the membrane itself. All right, let's now take a look at a typical plant cell, which are almost identical to animal cells. They contain most of the same organelles, most of the same structures, but there are some important differences. Who would like to tell me what one of those important differences are?
Cell wall.
Cell wall, correct. So animal cells lack cell walls. Animal cells are only enclosed by a plasma membrane. Plant cells also have a plasma membrane, of course, all cells do, but they also have an additional layer called the cell wall. Correct. Another difference between plant and animal cells.
Chloroplasts.
Chloroplasts, correct. Here we have organelles called chloroplasts. Yes, which you don't find in animal cells. And one other major difference, something that you can see here, which you do find in animal cells, but very, very few, and they're very, very small.
Vacuole?
Vacuole, correct. Plant cells tend to be characterised by this enormous vacuole, which occupies often almost the entire space within the cell. All right, so there we have it, central vacuole. As I said, you do find sometimes very small vacuoles in animal cells, but certainly nothing like this. All right, and then we have other structures which are the same in animal cells. Nucleus, endoplasmic reticulum, Golgi apparatus, mitochondria, and so on. All right, let's take a look at the nucleus. The nucleus is an organelle. It is an organelle because it's enclosed, it's surrounded by a membrane, but not just one membrane, it's surrounded by a double membrane, and that double membrane is called the nuclear envelope. The nuclear envelope is continuous with the membrane of another organelle called the endoplasmic reticulum, so they are always associated together. The nuclear envelope is perforated by these tiny pores called nuclear pores. And the pores aren't just little holes, they're actually composed of protein complexes. And the nuclear pores will allow the passage of some molecules to pass from the nucleus into the cytosol of the cell, or vice versa. All right, let's look at that here. So here's our typical cell, and you can see in blue the nucleus. Let's enlarge the nucleus. This is what we're looking at here, and it's been cut away. We can see on the outside of the nuclear envelope the presence of all these pores here. Inside we see this dark staining region, and this is called the nucleolus. Here's one nucleolus here, but there's also lots of small nucleoli within this cell. And you probably saw this in prac. Did anyone see nucleus and or a nucleolus in your prac class?
No, I didn't.
No. What about when? Sorry?
Didn't get that zoom in that far, but yeah.
Oh, right. Okay. All right. You didn't.
I didn't anyway. Maybe someone else did.
Right. Okay. That's okay.
Managed to see a nucleus, but not more than that.
Yeah. No. It's with a standard compound light microscope. You could probably see. You might see the actual nucleus itself. More likely, you would see the nucleolus. This dark region here. But yeah, difficult to see any of the other organelles within a cell. All right, and down here we can see that nuclear envelope. So remember, two membranes. So here's one lipid bilayer, so that's one membrane, and here's another lipid bilayer. So double membrane, and here's the nuclear pore complex. All right, the nucleus contains the DNA of the cell. It houses the DNA. The DNA is never found in the cytosol of the cell. It's always found in the nucleus. Although I must say there is a tiny bit of DNA in a couple of the organelles. We'll come back to that. DNA molecules are found in the nucleus, wound around proteins called histones. And when the DNA winds around these histone proteins, it forms what are called nucleosomes. Let's look at that. So here we have in blue the strand of DNA winding around these histone proteins. And here we have what's called a nucleosome. When a cell is not dividing, it's not undergoing cell division to produce new cells, then the DNA is referred to as chromatin. And in this form, you can't actually see the DNA using your standard compound light microscope. It's just not visible. In the form of chromatin, which is illustrated here, some of the chromatin is very tightly condensed. It's very tightly aggregated, and we refer to this part of the chromatin as heterochromatin. Other parts of the chromatin is more dispersed, and that's referred to as euchromatin. You certainly don't need to remember this. That's just a typical part of cell biology that's traditionally taught. All right, now when the cell is actually preparing to divide, into new cells, and we'll talk all about the different types of cell division in second semester, then the chromatin strands will start to condense. And when they condense ready for cell division, they appear magically under the microscope. This is what you'll see. Here we have a cell. This is a plant cell that's getting ready to divide, and you can now clearly see the DNA. And when it's visible like this, we refer to it as chromosomes. And this is what they would look like. Each pair of, we've got a pair of chromosomes here and here. This is what they would look like if you used a really high-powered electron microscope. Okay, but this is what you would see if you actually prepared a really good sample of a cell that was about to divide. All right, so we've seen that the nucleus contains one or more nucleoli, singular nucleolus. So these are the dark staining regions that we can see in this image here. And the reason that they're so densely staining is because they're very, very concentrated regions of DNA, RNA, and protein. So there's a lot going on in those regions within the nucleus. The nucleolus size and the number of nucleoli really depends on how active that particular cell is. The nucleolus is the site of the synthesis of a particular type of RNA molecule called ribosomal RNA. You don't need to remember that now. We'll talk about different types of RNA molecules later on. But this is where ribosomal RNA is synthesized, and it's also the site of assembly of another cell structure called the ribosome. This is where they're put together, or rather components of the ribosome are actually put together within the nucleolus. All right, and talking about ribosomes, here we are. So ribosomes are structures found in both eukaryotic and prokaryotic cells. They're not termed organelles because they're not enclosed within membranes. What are ribosomes made of? They're made of several different types of proteins, and they're made of a couple of different types of RNA called ribosomal RNA. So these ribosomes, or rather the subunits of the ribosomes, are assembled within that nucleolus region in the nucleus. Then those subunits, there's two of them, 2 subunits per ribosome. They're actually transported then out of the nucleus via the nuclear pores into the cytosol of the cell. Remember the fluid interior of the cell. And here the 2 subunits associate with each other to form a functional ribosome, which is what we're seeing here. Here the ribosome does its job. It attaches itself to a different type of RNA molecule called the messenger RNA. It moves along this messenger RNA molecule and it decodes it. It reads the code found within the messenger RNA, and it translates that code into a polypeptide, which we can see that's being formed here. So ribosomes are often referred to as the protein factories of the cell. These are the structures that make proteins, all the proteins that are required by the actual cell. Sometimes you'll have several ribosomes binding to the same strand of messenger RNA. And when this happens, this is referred to as a polyribosome or a polysome. So where do we find ribosomes? The ribosomes may be found free in the cytosol of the cell, or they may be attached to an organelle called the endoplasmic reticulum. All right, let's consider now what we call the endomembrane system. So we know that the cell is enclosed in a membrane, which we call the plasma membrane. We know the nucleus is enclosed in a membrane, a double membrane called the nuclear envelope. And of course, membranes also enclose other structures inside the cell, and collectively they're referred to as the endomembrane system. So for example, structures or components or organelles, if you like, such as the endoplasmic reticulum, the Golgi apparatus, the lysosomes, endosomes, and vacuoles. These are all enclosed in biological membranes. All right, who needs a 5-minute break?
Yes, please.
Yes,
I do.
please.
Oh,
Yep, all right, I'll see you in 5 minutes.
okay.
Sorry? Okay, is everyone ready to go?
Yes.
Yes. Yes, Liz, Justin, Brad, Andrew, are you back?
Yeah, hi.
Hi.
G'day,
Who's that?
Justin.
Oh, hello, Justin. Sorry, I couldn't see the mic there. Okay. Liz, are you back? All right. Okay, that's all right, Brad. No problem. All right, Liz is back. All right, no worries, Brad. All right, our next organelle, the endoplasmic reticulum, abbreviated to ER, extends throughout the cytosol. It consists of a network of sacs, membranous sacs called cisternae, and it's continuous with the outer membrane of the nuclear envelope. So remember the membrane of the nucleus. The cisternae are flat and sheet-like, and they cover an extensive surface area. Often they comprise more than half of the total membrane area of the cell. Let's look at the cell up here, and in brown you can see how extensive the endoplasmic reticulum is, and it's clustered around the nucleus of the cell. So here it is enlarged, and you can see these flat membranes, the cisternae, and the endoplasmic reticulum has two different types that are labelled here. So this portion here, which is closest to the actual nucleus, is referred to as the rough ER, the rough endoplasmic reticulum, and that's because it has this rough appearance. That rough appearance is due to many ribosomes being attached to the outer membrane of the ER. Now, this front portion here of the ER is referred to as smooth endoplasmic reticulum because it lacks that association with the ribosomes. Now, you'll see here these tiny little spherical structures, or they should be spherical, only half of them is shown. These are little bits of membrane that bud off from the endoplasmic reticulum. They're called vesicles, and they bud off containing some cargo, which is destined to go somewhere in the cell. I'll come back to those vesicles in just a minute. But let's enlarge this little portion here of the rough endoplasmic reticulum, and that's what we're looking at here. So we can see the lipid bilayer of the membrane of the ER. Here we can see some of those transmembrane proteins embedded in the lipid bilayer. And here we have the ribosomes that are attached to the rough ER. In fact, there's several ribosomes that are actually attached and moving along this one messenger RNA molecule. And they're decoding the message that's found in that messenger RNA, and while they're decoding it, they're synthesizing polypeptides, which are passed directly into the lumen of the endoplasmic reticulum. And then once they're in the lumen, then the polypeptides are folded with the assistance of folding proteins, and they're modified in some way. So for example, they might have sugar molecules attached, or they might be modified in other ways, chemically modified. So we've seen that there are ribosomes bound to the surface of the rough endoplasmic reticulum. Polypeptides are modified and folded within the rough ER. We saw that the polypeptides are passed from the ribosomes, which are making them, into the lumen of the ER, and there they're folded and modified. The smooth endoplasmic reticulum lacks that association with the ribosomes. The smooth ER predominantly functions to synthesize all the lipids required for the cell. The enzymes involved in lipid synthesis are on the cytosol face of the membrane. And the smooth ER also possesses enzymes that are involved in detoxifying lipid-soluble chemicals and also other harmful metabolic products. Our next organelle is the Golgi complex or Golgi apparatus. It looks very similar to the endoplasmic reticulum, but it's much smaller. The Golgi comprises stacks of membranes called cisternae, and the number of stacks varies from approximately 4 to 10. Although the Golgi apparatus that you would find in a small algal cell might only have 1 stack of cisternae. Golgi apparatus has a distinct orientation. One side is called the cis face, and this side of the Golgi apparatus always faces towards the endoplasmic reticulum. The other side of the Golgi is called the transface, and it always faces towards the plasma membrane of the cell. Let's look at that on the next slide. So once again, here's our cell, and in purple, you can see an example of the Golgi apparatus. Here it is enlarged. Let's look at the 2 sides. This side here is called the cis face or receiving side, and that's because it receives these little vesicles that are containing some cargo from the endoplasmic reticulum. So these vesicles are basically tiny little bits of membrane that have budded off from the endoplasmic reticulum, and then they're transported to this side, the cis side of the Golgi, where they then fuse, these little bits of membrane fuse with the Golgi apparatus and deposit their contents into the lumen of the Golgi. And then those substances are further processed as they pass through the Golgi apparatus. They're sorted, they're tagged in some way depending on what the substance is. Then you'll have these little bits of membrane that bud off from the Golgi. You can see here, the membrane here is budding off, and then it pinches off to form another little vesicle. And then that vesicle with its cargo comes off from this side of the Golgi, which is called the transface or shipping side. And these vesicles will actually be transported to different parts of the cell depending on their contents. So that's the Golgi apparatus. And here's a really nice image taken with an electron microscope. And you can see here the structure of the Golgi apparatus. Here's another view of the Golgi, and it's surrounded by these little transport vesicles. All right, so the Golgi apparatus processes and packages glycoproteins and polysaccharides. We've looked at the cis face or the receiving side of the Golgi, and here we have proteins and glycoproteins and so on entering from the endoplasmic reticulum via those little vesicles. Then they're modified, the molecules are modified as they pass through the cisternae of the Golgi. At the transface, this is where the products are sorted, packaged, and then released once again via these vesicles. All right, let's look at that together. part of the endoplasmic reticulum here, and it's showing those little bits of membranes budding off to form vesicles, which are transported to the cis face of the Golgi. Here we have the transface and a little bit of membrane which actually has these markers on it, which will determine where that vesicle, once it buds off, where it's actually destined for within the cell. So let's look at where some of these vesicles end up. Here we have a rather large vesicle, shouldn't be that large, but this one might be destined for the plasma membrane itself, where it will deposit its contents onto the actual inside of the plasma membrane. So this could be repairing a membrane and carrying, for example, proteins or other substances that are required to repair or maintain the plasma membrane. Here's another vesicle that could be carrying something that has to be secreted from the cell. So this vesicle will approach the plasma membrane and then it will fuse with the plasma membrane. In other words, we're adding a little bit more membrane material to the plasma membrane itself, and then the contents are deposited to the outside of the cell. So we would call this a secretory type vesicle. Now you can imagine that this is happening all the time. All these little bits of membrane that are coming from the vesicle are being added to the plasma membrane. So that means the plasma membrane is getting larger and larger all the time. This is balanced by the opposite process. That is, the plasma membrane can start to buckle inwards at any point. This is called invagination. And it can actually take in solutes and other substances from outside the cell into the cell. Then this will actually pinch off and move into the cell. All right, so the import of substances into the cell is called endocytosis, whereas the export of substances, as we saw down here, is called exocytosis. And of course, these two processes balance each other out. Now, here's another example of where a vesicle that buds off from the Golgi might end up. It might actually go towards another organelle, which is called a lysosome. This one's labelled here as an endolysosome. That's just an immature form of the lysosome. And this this vesicle might deposit its contents into the lysosome, because the lysosome contains lots of different enzymes. So that might be the contents that are being deposited there. So just some examples of where those products might end up. All right, and that brings us to the organelle called the lysosome. Lysosomes contain lots of different hydrolytic enzymes. These are enzymes that will break stuff down. They'll break down old organelles. They will break down large molecules into small molecules. So, for example, large proteins might be broken down into amino acids, which can then be reused. Large polysaccharides could be broken down into monosaccharides. RNA molecules that are no longer required can be broken down into nucleotides, and so on. The enzymes that are found within the lysosomes are manufactured in the Golgi apparatus. That's where they come from. And another important point here is that the interior of the lysosome actually has a very, very low pH in contrast to the rest of the cell. And that's because these enzymes, these hydrolytic enzymes, actually function at their optimum in an acidic environment. So the pH within the lysosome is very acidic, about 4.5 to 5, and contrast that to the pH within the cytosol of the cell, which is about generally kept at about 7.2. Now, the way that lysosomes maintain that very low pH is to actively take up protons. And you know from your chemistry lectures that if we increase the protons, the number of protons, then we're increasing the concentration of what ion? Hydronium, correct. If we increase the concentration of our hydronium ion, then we will decrease the pH. That is, it becomes more acidic. Okay. So there are our lysosomes. All right, we've looked at those little transport vesicles. And we've also considered these terms, these processes: exocytosis, exportation, which means exportation of substances, and endocytosis, importation. Our next organelle, the mitochondria. Mitochondria are referred to as the powerhouse of the cell. This is where energy is extracted from bonds, covalent bonds in carbohydrates and covalent bonds in fats, for example, and that energy is converted into useful energy in the form of ATP. So mitochondria are the site of cell respiration. Let's look at this. Mitochondria are thought to have evolved from engulfed prokaryotes. Very interesting theory. It's called the theory of endosymbiosis. And we're going to look at a little bit of the evidence that there is to support this particular theory. We won't go into the theory, but you can certainly look that up if you're interested. The theory of endosymbiosis. In a nutshell, what it proposes is that we had a pre-existing cell which engulfed another cell, or this second cell may have entered that first cell somehow. Then this cell within the cell, this survives, but not only does it survive, but it becomes highly specialised to carry out a certain function. And that's how it's believed, or that's where they believe that mitochondria and also chloroplasts originated from, that they were originally their own, they were originally individual cells that ended up just being organelles, basically, highly specialised organelles. All right, so let's look at mitochondria. Cells with a high level of metabolic activity tend to have lots of mitochondria. So if we think about liver cells, heart cells, for example, they can have anywhere upwards of 1,000 mitochondria per cell. All right, so here is one mitochondrion within a what looks like a plant cell. The structure of mitochondria. They have a double membrane, just like the nucleus. The outer membrane is highly permeable to ions and small molecules, and this is because the outer membrane contains lots of transport channels, that is, those transmembrane proteins. The inner membrane, on the other hand, is highly impermeable. That is, there's very few transport proteins embedded in the inner membrane. So this difference between those two membranes enables an electrochemical gradient to be set up. That is, an electrical gradient and a chemical gradient. And this gradient is crucial for enabling the extraction of energy from fuel molecules and the synthesis of ATP. But we'll talk a lot more about that in second semester. Let's look at the structure of mitochondrion down here. So we can see the outer mitochondrial membrane, the inner membrane that is folded into what are called cristae, singular crista, and embedded in that inner membrane are all the enzyme complexes that are required to synthesize ATP. Does anyone remember from a previous lecture we talked about the electron transport chain? Does anyone remember that? Yes, this is where it happens. This is where the electron transport chain happens within the mitochondria on this inner membrane. And I think I've forgotten something. Yes, here we go, mitochondrial matrix. So the aqueous solution within the mitochondria is referred to as the matrix. Okay, let's recap. Inner membrane of mitochondria is folded into cristae. This membrane is lined with enzyme complexes required for the synthesis of ATP. The matrix, the mitochondrial matrix, contains, and here is the evidence that supports the theory of endosymbiosis. The matrix space inside the mitochondria contains its own ribosomes. So normally ribosomes are only found within the cytosol of the cell. But here we have an organelle that has its own functional ribosomes. Not only that, these ribosomes more closely resemble the ribosomes found in a prokaryotic cell, such as a bacteria, rather than eukaryotic ribosomes, which have different structures. Within the mitochondrial matrix, you will also find a tiny amount of DNA. Again, very unusual. Most of the DNA of the cell is found within the nucleus, but the mitochondria has a small amount of its own functional DNA. Also very unusual is the fact that this DNA is circular, just like you would find in a bacterial cell. DNA that you normally find in the nucleus of a eukaryotic cell is not circular. It's always linear DNA. So a little bit of evidence there to support that theory of endosymbiosis. All right, our next group of organelles are called plastids. There's a few different types. We're only going to focus on one type of plastid that occurs in plant cells and in some protists, for example, algae. These are photosynthetic organelles called chloroplasts. They very closely resemble mitochondria in structure in that they have a double membrane. Each of those membranes has a different permeability. Chloroplasts also contain some ribosomes. They also contain a small amount of circular DNA. So once again, chloroplasts are believed to have evolved from engulfed prokaryotes. But this time, these prokaryotes have the ability to carry out photosynthesis. Chloroplasts contain light-absorbing pigments, mostly in the form of chlorophyll, but there are different types of pigments. They contain a third set of membranes, an internal set of membranes, which are referred to as thylakoid membranes. This is where the photosynthesis takes place, or rather part of photosynthesis. In places, the thylakoids are stacked on top of each other, and then they're referred to as grana. Chlorophyll molecules are actually found embedded within the thylakoid membrane. All right, here we have a plant cell, and in green, of course, we have the chloroplast organelles. Let's look at this representation here of one chloroplast with the outer membrane, and then just within that, the inner membrane, and then to the inside, that third set of membranes, and these are called thylakoid membranes. They contain the pigments and protein and enzyme complexes involved in photosynthesis. Here we can clearly see these stacks of thylakoid membranes. Each one is called a granum. The fluid interior of the chloroplast is referred to as the stroma. Our next organelle, the microbody. Microbodies remove unwanted compounds from cells. They're very similar to lysosomes, except that rather than containing hydrolytic enzymes, they contain oxidative enzymes. For this reason, microbodies actually function at a pH closer to neutral. So that's a main difference between microbodies and lysosomes. Remember, lysosomes enjoy that very low pH, and hence the requirement often to compartmentalise these functions within the cell because of the different requirements. All right, oxidative enzymes obviously catalyse the oxidation of a substance. What do we mean by oxidation? Let's revise. Who can give me a very brief definition of oxidation? Oxidation. So we need 1, 2, 3.
Addition of an electron.
Lost, right?
Loss of electrons. Yep. Loss, that's all right. That's why we're revising. Loss of electrons. Yep. What about gain of oxygen? Gain of, oh dear. That's supposed to say gain of oxygen. Or loss of hydrogen. Loss of hydrogen. All right, so in this case, the oxidative enzymes catalyse the removal of hydrogen from molecules. That's how they start to disassemble these molecules or inactivate them, if you like. And they couple that hydrogen to oxygen to form hydrogen peroxide, H2O2. Now, hydrogen peroxide itself can be damaging to cell components. So it's good that they're contained within the microbodies. And the microbodies actually contain a very, very high amount of this enzyme called catalase. And catalase will catalyse the breakdown of hydrogen peroxide into harmless products, water and oxygen. And you'll remember maybe that if you see ASE, then you know you're looking at the name of an enzyme. Not all enzymes end with ASE, but most of them do. All right, we have two examples of microbodies here, peroxisomes, which function primarily to oxidize amino acids, and glyoxysomes is another type. So there's a few different types of microbodies. Here we have a really nice image of some plant cells, and these plant cells have been visualised using a fluorescence microscope where different parts of the cell are tagged with different fluorescent molecules, so you can actually see them as different colours. So in red we have chloroplasts, and in green, these little green spots here represents peroxisomes. So they tend to associate very closely with chloroplasts. Okay, nearly there. All right, now let's look briefly at the skeleton within the cell, which is called the cytoskeleton. Cytoskeleton imposes and maintains structure of the cell. It actually does a lot of things. It fixes organelles, it moves organelles, it maintains and remodels cell shape, it allows cells to move, some cells to move. The cytoskeleton is crucial for the process of cell division and so on. There are three major elements of the cytoskeleton: microtubules, microfilaments, and intermediate filaments. And we can see part of a cell here, and we can see those three elements. So this structure here is the, where's the label? Here, intermediate filaments, and these blue tube-like structures here are the microtubules, and then these green tube-like components of the cytoskeleton are the microfilaments. And you can see how some of them are attached to mitochondrion, and how they're also attached to the endoplasmic reticulum. All right, so let's start with microfilaments. Structure of microfilaments: they have a diameter of 7 to 8 nanometers, and they're composed mostly of a protein called actin. Actin proteins will attach to each other to form these long chains. And the chains, more than one chain will associate with another chain to form a microfilament. Interactions between actin proteins and myosin microfilaments are the basis of many organelle and cell movements. Microtubules structure: diameter of microtubules 25 nanometers, and microtubules are composed mostly of two different types of protein: alpha tubulin and beta tubulin. We can see that here. So we'll have here's the two proteins, alpha and beta. So we have an alpha associating with a beta, alpha, beta, and so on to form this long chain, which is called a protofilament. You don't need to remember all this. And then we have several protofilaments attaching to each other to form this tube-like structure, and this is referred to as the microtubule with the diameter of 25 nanometers. Microtubules take on different structures, which may be radiating, bundled, or parallel. Let's look at those on the next slide. All right, here's an example of radiating microtubules. Here we have an example of parallel microtubules as seen in the axon of a nerve cell. And here we can see bundled microtubules in plant cells. Microtubules also play a very important role in cell division. We can see here in green, they're actually forming a structure called the mitotic spindle. And without this structure, cells cannot divide. In red, we can see the actual chromosomes, which are being attached to the microtubules before the cell divides into two cells. And finally, intermediate filaments, the third major element of the cytoskeleton. These are really tough, these filaments. They have a diameter of 8 to 10 nanometers, and they're composed of several different types of proteins. They function primarily to provide mechanical support for both the cell and for the nucleus itself. Here are some examples of intermediate filaments. Keratin, you're probably familiar with, really tough protein found within nails, hair, and so on. Here's another example. Nuclear lamins are also intermediate filaments, and they're shown here within the actual nucleus of a cell. So they basically provide that structural support within the nucleus, and they're also known to attach to the DNA. All right, finally, we move on to prokaryotic cells. So remember, when we talk about prokaryotic cells, we're referring to bacteria and archaea. Prokaryotic cells, or most of them, not all of them, but most of them, are surrounded by a semi-rigid cell wall. So this cell wall is to the outside, of course, of the plasma membrane. Prokaryotic cells lack membrane-bound organelles. So they don't have a nucleus, they don't have endoplasmic reticulum, Golgi apparatus, lysosomes, and so on and so on. They do contain other structures, but not membrane-enclosed structures. They don't contain a nucleus, but prokaryotic cells, of course, have DNA. All living organisms contain DNA. But prokaryotic cells have mostly circular DNA rather than linear DNA that you find in eukaryotic cells. The DNA found in prokaryotic cells just sits in the cytosol, not within a nucleus. Okay, so you might recall that we referred to mitochondria as the powerhouse of the eukaryotic cell. So of course this is where ATP is synthesized. Prokaryotic cells also have to make ATP. That's quite universal, that requirement. But because they lack mitochondria, organelles called mitochondria, where do they actually set up that electrochemical gradient that's required to produce ATP? The enzymes involved in this process are found on the plasma membrane of a prokaryotic cell because, of course, they lack mitochondria. Some prokaryotes can photosynthesize, just like plants and just like algae, but they don't contain organelles, which means they don't have chloroplasts. So those pigments that are involved in of trapping that energy from light are actually found on the plasma membrane. Very unique to some prokaryotes is the presence of what's called a flagellum or flagella. These are rotating structures that enable the prokaryotes to move. And we can see that on the following slide here. So this is a nice representation of a prokaryotic cell, an external cell wall. To the inside of that, in yellow, we have the plasma membrane. To the inside, we don't have any organelles, but we can see lots of little ribosomes. And here we have the DNA, which is not enclosed within a nucleus. Okay, here we have an example of a flagellum. Looks like a tail, but it's not. It's actually surrounded by or enclosed within plasma membrane, and it can actually rotate. It can spin quite fast and enable the prokaryotic cells that have these to move along. And they're often used to identify different species of prokaryotes, because some prokaryotes only have one, some have many, some have them at one end, some have them from the side of the cell, and so on. And that completes our lecture on functioning cells. Thank you everyone for your attention.
Yeah, all right, that's it.
You did well. That's a lot to get through. All right, so this week's tutorial questions. So now you can complete tutorial 7 on solutions, not assessed, but as you know, very important nevertheless. Some more practice for you, so make sure that you go through these calculations here. And remember, you can check your answers via the online quiz, which doesn't close, through the non-assessed online quiz. And also for this week, another non-assessed set of questions on functioning cells. So once again, make sure you check your answers, and please let me know if there's any that you don't quite understand. If you get one incorrect, let me know. All right, so do you have any questions for me?
No. You good?
No?
No, not me.
No? All right, is everyone, everyone's aware that you need to write 2 prac reports by the end of semester? Yes? Yep, good. 2 prac reports. One must be on the acid and base titration prac, and the other report you can choose any of the pracs that you did to write that report on. I normally recommend to students that once you've written the first report, just to email that to me as a draft, and I can check that for you and give you feedback before you actually submit that and before you complete the second report. Okay, so keep that in mind. Obviously, I need that draft as early as possible, at least 1 week before it's due, to make sure that I've got time to actually look at that and give you some feedback on it. Okay.
Awesome.
Sylvan, was there one prac that was recommended we don't do?
That you don't do?
Vaguely recall that, but I could be wrong.
Yeah, I can't. Yeah, I think there is, and I can't remember. So let me run me through the pracs you did. You did cell membranes. That's an excellent one to write up.
Yep.
You did prokaryotic and eukaryotic cells.
Correct.
That's a good one to write up. Acid-base titration, you have to write that one up. You also did,
Concentration curve.
yes, the standard curve, determination of concentration. Yes, that one you can write up.
Was it the first one on Sunday?
What was the first? Was it just a microscope prac?
Yeah, that was the
Yeah,
one.
it's just
Yeah.
a microscope prac.
That's just learning how to use the microscope. No, don't write that one. Yeah, that one's very difficult to write up.
Thank you.
Because really think about that prac in terms of the results. You really, all you're doing is calibrating the microscope, filling out the table. There's not a great deal that you can actually discuss.
Okay, thank you.
Yeah. So, yeah, stay away from that one. Some students do choose to write it up, but it is a difficult one, I think, to write up properly. So basically, any of the other pracs that you did, you can choose to write up, depending on what interests you the most. I think cell membranes is a fantastic prac to write up. There's just so much that you can write about in terms of analysing your results and explaining what you expect to see, what you did get, and why you think you got that result.
Okay.
Yeah, I think that's a really good one. Yeah. All right, if there's no other questions, I'm going to have dinner.
Enjoy.
What happened?
Yes, thank you. You too. You too. All right. Good night, everyone.
Good night.
Right.